So let's start with the nature and the scope of the problem of bacterial genomes. So bacterial genomes are gene rich. Genes cover 85 or 90% of bacterial genomes. And for this reason, you can actually just use shared gene content as a proxy for genome-wide similarity. And that's quite handy. It's a kind of language that's used in the microbiology community or certain parts of it. And it allows us to, it's just a cheap way of being able to talk about how similar different genomes are. But we're not, but it really does incorporate most of the genome. So what's interesting about bacteria, so I'm going to take E. coli as an example, that if I took two random E. coli, they might only share 50 to 70% of their genes and therefore their genomes. I'll talk about how this happens exactly in a minute, but for comparison, obviously, if you ask that about two human genomes, then I mean, the nearest equivalent would be asking how much is alignable, and that's over 99%. So you do have big events happening in human genomes, but human genomes are enormous compared to bacterial genomes. So, and as a proportion of the genome, they're structurally very, very variable. And I think a thing that a pervading figure throughout this talk is this canonical gene frequency curve. So this seems to be a universal pattern that's seen across bacteria. You can actually see it in between genera in a different way, but within a species, this plot is a histogram. I've taken all the genes, or Marie Touchon has taken all the genes within just 20 E. coli genomes and then plotted the frequency of them. In other words, this peak on the left is the genes that are only seen in one genome, these in two, these in three, and these are only 11% of the genes are actually seen in all of the genomes. And an immediate consequence of this is it's very hard to choose one genome which is going to allow you to access all of that. So another interesting factor about these things is that genes kind of appear and disappear in blocks. So this is a plot showing this is a single position in multiple E. coli genomes, and these blocks are modules containing multiple genes. And so what you can see is across these genomes, this is the same position. You've got basically a random assortment of mosaic of these different blocks of genes. So the question for today's talk is, how can we cope with these chunks of genome and still do SNP calling in bacteria? I want to be able to discover all the SNPs in a set of genomes in an effective way. And this talk is all going to be about doing this from sequencing reads, but the problem remains even if you have perfect assemblies. Even if you have all those, 20 or 100 or 1,000 perfect bacterial genomes, how do you address all of the SNPs between them when there's so much rearrangement going on? So here it is as a cartoon. These vertical regions are different E. coli genomes, let's say. The colors are different genes or intergenic regions, and the numbers are SNPs. So here, these blue genomes, these blue genes contain 4 SNPs between them. These browns contain those SNPs and so on. And I want to know how many of those I can discover. I want to discover all of them if I can. And if I just chose one of them as a reference, then I'm not going to belabor this too much, but if I just chose one of these as a reference, obviously I can only access the variation that's inside that reference. So in this toy example, I've calculated the numbers of the percentage of the genomes, of the SNPs that you can find using each of those genomes as a reference. Now this is a toy, but that's what we expect to see this in reality too. I've got a comment in there that we would see this in a null model. I've actually removed the null model from the slides. The question is, what really happens empirically? So we're going to address that. I'm going to give us one bit of terminology, which is quite handy. I'm going to call hard reference bias this problem of having huge chunks of genome that are missing from any single reference. So in other words, it doesn't matter what reference you choose, there's going to be chunks of genome missing and you're not going to be able to find stuff. That's inescapable. That's part of the biology. And that's because of this property of bacterial genomes. Soft reference bias is the decreasing ability to map and call variants as the edit distance between the sample that you're studying and the reference increases. And that's the usual argument we use when we're talking about reference graphs, graph genomes. So basically, the more different your read is from the genome, because it's come from a diverged sample, diverged genome, the more different it is, the harder it is to match, the harder it is to be confident that it goes where you're putting it, And then when you're doing variant calling, you get more and more artifacts to do with alignment. Okay, so I'm going to talk about, I've realized it's about 8 person-years of work from 3 incredible people, Rachel, Michael, and Leandro. Rachel developed Pandora. Michael had joined like 2 years ago, and he's added all the de novo assembly components and a lot of the evaluation. And Leandro is our software developer who's basically revolutionized the efficiency of the whole thing. And this is what we're going to do. I'm going to start with some spoilers telling you what we're going to do. And the reason I'm going to do that is to motivate our definitions in this following section. I'm going to talk about the representation of the genomes and how we're going to represent our graph. It's going to be a graph of, it's a directed acyclic graph of minimizers. And then the next section will be how to infer the genome of a single sample. So I've got reads from one genome, and I've got that graph, and I want to infer that genome as best I can. And we do it in a 2-stage approximation. Then having done that, say I've done that independently for a bunch of samples and I want to compare them, what's the best way to compare them? That's the 3rd section. Okay, so the strategy is this, or the motivation is this. Genomes evolve by recombination and mutation, okay? So new samples should look like noisy mosaics of the old ones. If I've seen 1,000 genomes before and I look at a new one, simply because of their shared ancestry and the dominating forces being just recombination and mutation, new things should look like spotty mosaics of the old ones. So that motivates us to try storing a reference panel of genomes in such a way that when you can map to that panel instead of a single reference. And when you map to that panel, you want to allow reads to map across recombinations. Okay, that's the first thing. The second thing is, like, when you've mapped it, how do you infer a single genome? And we're going to do that by finding the closest mosaic through that reference panel to the unknown genome. That's the first step. So our first approximation is a mosaic in our graph. And then that's not going to work completely. So we'll fix up all the errors with local assembly. And we go through that whole process without ever having to do full graph alignment, which is quite a costly process. Okay, here we go. Into the blue, that's representation. So we call a pan-RG, a pan-genome reference graph. It's an unaltered set of DAGs, okay? Each graph, each DAG represents some orthology block. It will be a gene or an intergenic region. We've used genes and intergenic regions in this talk, but you could use mobile elements or other things. And we completely let go of higher order structure. We don't know anything about operons, gene order, the whole ordering of everything in the genome. We're not going to touch that for now. And then each local graph is roughly speaking like a multiple sequence alignment. So if this is an alignment of different genomes, and if you lay them out in a graph, I'm sure you've seen things like this before, it gives you a representation that shows you that everybody shares this ATC at the start, this GGA at the end, and you can split off either like this or like that. Now, once you've got that, you're going to want to enable mapping and search, depending how you think about it. So typically you want to index this graph, and Paola Bonizzoni discussed this yesterday in detail. And the problem she brought up is exactly the thing that I have to talk about first, which is blow up. So if I have a reference and I've got a VCF and I've got 10 SNPs all very close together, and I've built this from, I don't know, 100 genomes, and I want to build my graph. If I allow 10 independent bubbles, 10 independent SNPs in my graph, then when I index it, I'm going to have to cope with 2 to the 10 different paths through those things, okay? But I've only ever seen about 100. So indexing is actually critical for mapping, but this blow-up problem is a real issue. So if you're starting from this position of having a reference and some variants, there are a bunch of different tools, GCSA, Bubble, I don't know how you pronounce Bubble, VBWT and VG. They have different approaches to try and avoid blow up. You can either use heuristics to sort of clean up the graph. You can just take in variants based on the frequency, which is, you know, it's quite a rational approach. You could index only a subgraph, which you hope will be good enough. You could index, there's a really interesting idea from Ali Ghaffari and Tobias Marshall to just index paths supported by the reads. So obviously you have to re-index with every read set you're analyzing, but that's quite clever. Or you could see it as an optimization problem. Ben Langreid's team developed Forge, which you can optimize in terms of sort of frequency or trying to avoid adding too much repetitiveness to your graph. So all these we basically developed all kinds of approaches to try and get rid of this problem. And the second option, of course, is you can add a second data structure, and that indexes known haplotypes. So that acknowledges that there are only a small number of haplotypes that really exist through this labyrinth. So what I'm going to suggest is that blow-up is partially a consequence of an unrealistic model. So if you have a linear reference, just a linear reference, it's a simple model, and it's a very strong prior. You expect everybody to look like the reference, mostly. So a more realistic, or perhaps a more complicated model, is to say every genome is a recombinant of things I've seen before. If you take your graph, which is made from a reference and all your SNPs, and you allow all possible paths to it, you're basically saying you're allowing an infinite recombination rate, because you can believe that recombination could have happened everywhere. And that is a very extreme position, okay? That's the worst possible scenario. It's not as bad as saying you can have every base at every position, but it is extreme. So if you take a less extreme model, blow-up reduces. So what we do is we've got, I've just made up this name, the recursive cluster and collapse algorithm, which is used to build a DAG from multiple sequence alignment. So it allows nesting, and by nesting, I mean allowing variants on top of variants. So you could have a SNP on top of a long haplotype, or you might have SNPs underneath a long deletion. And it avoids blow-up, and it does this by preserving existing haplotypes until you meet a sufficiently long non-varying region. Okay, so how does this work? I'm going to do this very visually. Here's a multiple sequence alignment. I'm sure that's a very natural image for you. So you can imagine that all the rows inside this rectangle are sequences. They're multiple sequence aligned. And what I do is, the first thing I do is I look for vertical slices of length at least k, some parameter, where all of my rows are identical. So inside the left-hand yellow section, all of these rows are identical, and the same over here. So having done that, from now on, all of those yellow regions are going to be collapsed. There's nothing going on there. So, and now the three blue regions are completely independent. Okay, my algorithm can sort of treat them independently. The next thing you need to do is for each blue block, use k-means clustering to reorder the rows. We actually convert the string to a sort of k-recurrence dictionary, and then you cluster to minimize the differences. And when you've clustered, you now have these clusters here, and then you repeat this recursively. So within each block, I now apply the same process I did before. I look for vertical matches within my blocks. And because they're now more similar, I've got more probability, more of a chance of finding vertical matches, and I keep going. And that ends up giving me, so here's I don't know if this is really helpful, but this is a little video of it going with some sequence. You find matches, you collapse, and you find more matches, and you collapse again, and you end up with a graph. So that's how we do it, okay? And in this complicated region, we've preserved those sequences that we know always go together. Having done that, I've got a DAG, but I want to sparsify it slightly. So what we do is we generalize the notion of a WK minimizer of a string to a DAG. So for all paths through this, we take all the k-mers starting in a given window of length W and choose the least under some ordering. And then that way you can replace the original DAG with a new DAG, but just of minimizers. And you can index that just as a map from the minimizer to a list of gene ID, so which of the local graphs we're talking about, and the position and the strand. So now we've got an index going from minimizers to all my different genes and the positions inside them. Okay. I'll remind you that I was just talking about representation there. We move into the next section, which is yellow, single sample inference. And you can, this is our workflow. We start with our reference graph, which we index. Each of these squares, rectangles, which are sort of drawn behind each other, are independent samples. For each sample, I can independently quasi-map the reads to it. I'll show you what I'm going to do with it. And then you end up noticing alleles that are not in the graph, and you send them back to the start, put them back inside the graph, and keep going. And once you've done all of this, you're then going to compare all the samples against each other. I'll explain all this in more detail. I'll just point out that this section in the middle I can parallelize by sample, because I'm mapping the reads of a sample to the graph. Samples are all independent. I augment the graph, and then my final section where I'm comparing everybody, I can't parallelize by sample because I want to compare them all, but I can parallelize by gene. So the whole system is very parallelizable. Okay, I want to talk about single sample inference. Reference-based, normal variant calling is basically a first order approximation. You assume the sample is equal to the reference, you map your reads, do a pile up, you look for, I mean, the simplest stories, you look for isolated SNPs, which are clear, they're obvious in the pile up, you fix them and you're done. This works really well when it works. It's really bad for indels, and it's a pain when there's clustered variation, but it does work. Our approach is 2 steps. First, you find the recombinant of the previous genomes that best approximates your sample, and then that's a good reference. And you know, there's a long history of doing this kind of thing. Corbin and Schneeberger introduced these kind of ideas in 2009, Alex Stilthe in the MHC, Serena Machuca in Plasmodium, and Danny Valenzuela in 2017 and 2018. So these ideas have been around. So what do? We quasi-map the reads. What do I mean by that? I sketch the read and collect its WK minimizers. My index tells me which genes have got matches and where. And for each k-mer shared between the read and some gene, I define a hit as the coordinates of the minimizer, both in the gene and in the read. And then you basically go through a filtering process to get rid of isolated hits, which look like errors, and look for clusters. This is easier in a figure. So what I've got there is 4 cartoon local graphs. They're different genes, got known variation in them. I sketch them into their k-mers. I take a read, sketch that into its minimizers, and then I look for clusters of shared minimizers between the local graphs and the read. So for example, I've got some red k-mers are errors. I want this to work for both Nanopore and Illumina, so I've got to be braced for errors. So these orange things match with this guy over here, these green things match with this guy over here. So I found inside this long Nanopore read, I found a bunch of genes. Now, having identified which genes are present along the read, we can discard the k-mers which are errors, the red guys before, and only after discarding the red ones do we increment counts on the k-mers in our genes. So this allows us to drop to a comparatively low k-mer, like 15 or something, without incurring a big cost of false repeats. And then once you've done this, I've got a DAG, I've got counts on it. I can use dynamic programming to assign the maximum likelihood path along here to estimate the genome. And I'm talking about haploid bacteria here. So it's not consensus, but it's the closest path in the DAG to the genome under study. Okay, where does that leave me? Well, I now have a mosaic, which is the closest path to my sample. Inevitably, there are going to be some SNPs and indels which are not in my graph. Okay, my graph is just built from some reference panel of genomes I started with. And it's not meant to be an archive of all known variation. So there will be gaps in coverage. So when you look at the mapping, this is an example of a mapping to a section of a gene. And inside these green areas, we've found regions where the blue line is the depth of coverage. There's a depth of coverage collapse. And actually, the red dotted lines show where there are actual SNPs, signaled by SNPs that are not in the graph. So what do? We locate these regions, the green regions. We slice out the bits of the reads that overlap them, and we do local assembly. And this is what the component that Michael has added. I'm not going to have time to detail the rather clever algorithm he's used for enumerating paths through here without blow up. We don't want to assemble a single path. All we want to do is get a good set of candidates. And then we're going to augment the graph with those candidates. And then we're going to do a second round of mapping to find the right one. So roughly speaking, what you have is you've got your reads. When you map them, these orange reads just don't map to this. There's something missing, but a local assembly finds it. We put it back in the graph, and when you remap, suddenly you've got more reads mapping. So that's that picture here would only ever happen in a bacteria if you've got either a multi-copy gene or a mixed infection. But anyway, you get the idea. And then we do genotyping. I'm not going to say much about that. It's just a Poisson model. We look at the coverage on the alternate alleles and called genotypes based on the coverage. And that gives you also gives you a quantitative measure of your confidence, like what's the likelihood of an A here versus a G. You compare those likelihoods in a sort of like a Bayes factor, and you get a confidence measure. I don't need to cover that again. And so, okay, what are we halfway through, more or less? What have we done so far? I've claimed that for bacteria, one reference genome is completely impossible. That's a bit dramatic, but it really has a problem with hard bias. Okay, it also has a problem with soft bias. Bacteria can have higher levels of diversity than humans at a per base level as well. Pandora generates a set of local graphs from multiple sequence alignments. Soft bias is addressed by using the graph. Hard bias is addressed by having a set of graphs. And for each sample, we find the closest mosaic, use local assembly to fix stuff, and augment the graph with everything that we found so far. Okay, that takes us through sections 1 and 2, and now into section 3, which is like how we're going to compare samples. So as soon as we compare genomes, we have a representation problem. What reference you use really affects how you describe a data set. So here I've got a red and a blue genome and a black reference genome. And the red and blue only differ by a SNP. But because I'm using black as a reference genome, I have to describe a variant which starts here, and I have this flanking sequence, which is shared by both of my alleles. Well, that's a pain. So it looks like this in a VCF. Now, if instead I had chosen the blue genome as a reference, then suddenly I just get a SNP. So what I'm saying is, having already discovered my variation, when I try and describe it to other people, my choice of reference here as a coordinate system really affects how interpretable the results are. So what we do, we've already inferred one mosaic approximation for each sample, the closest path. We wipe the graph clean, so to speak, and we draw on all of these mosaics, one for each sample. And we increment counts along the graph for wherever they go. So we can see which regions of the graph are shared by lots of genomes and which are rare. And then we use dynamic programming again to choose the most supported path. But here, what we're doing is we're choosing the best reference genome to describe all of those samples, all of those genomes. So the corollary of this is we now have a multi-sample VCF, or we now build a multi-sample VCF, which has got the best reference for describing this data set, because it gives you the most succinct description of the segregating variation between this set. Basically, you want more SNPs and less long alleles, or less artifactually long alleles. And it's very easy to annotate, and it's very easy to communicate with people. And there's a second part to this. There's strong reasons to support VCF in terms of community engagement and downstream tools and so on. As far as I know, this is the first time someone's used a graph tool to support VCF without making the user provide the reference genome in advance. If you provide a normal linear reference genome in advance, you inherit the hard reference bias of a single reference. So we don't want to do that. We want to choose a reference genome based on our data. Okay, I've basically gone through our methods. I'm now going to talk about what our results look like. Just going to remind you of the heroes here. Rachel, Michael, and Leandro I've talked about before. Leah is a microbiologist and a genomicist, joined us recently, and she's been working on Pandora and on using it sort of in real life, and she's been helping with this evaluation. And it really helps to have an expert biologist and bioinformatician in the team. So if we what we started to do is we built a pan-RG of E. coli. We took 20-odd thousand genomes from 350 REF-seq genomes. I'll come back to whether or not that was a good decision. And about 14,000 intergenic regions from actually a different 228 genomes. Now, basically because of that frequency distribution curve I described before, 59 or about 60% of the genes are actually linear. Okay, there's only one allele in those 350 genomes, and 44% of the intergenic regions are also linear. So there's always going to be a mix of sort of complex and easy graphs. And then I'm not really talking about implementation much here, but we took 20 gigs of RAM and 70 minutes to index that graph using 16 threads. And the reason it's that fast is thanks to Leandro. Okay, so I'm just going to remind you, we just want to find all these SNPs to evaluate how well we're doing. We want to see whether or not we can find all the SNPs. Okay, that sounds good. So we start with a data set for evaluating it. We took 20 E. coli from across the phylogeny. E. coli is quite a diverse species. It's quite recombinogenic, so it's got quite a big pangenome. And we made sure that we collected samples that had matched Illumina and nanopore data. Where possible, we tried to insist that the sequencing of Illumina and nanopore was from the same isolate. We got long read assemblies, we polished them, we remapped the Illumina reads to the assemblies, masked positions that looked weird, either because the Illumina reads didn't support the long read assembly. And so what that left us was with masked genomes with positions that we were sure were right. And we want to use those to find the true SNPs and indels between the genomes, because we need a set of truth and to see how well Pandora can discover them. So here's what we do. These are our genomes. This is the phylogeny of E. coli. I've colored different clades in different colors. Well, Leah has colored them. In E. coli world, those colors actually get are given what they're called phylotypes. And then our genomes are marked with these gray triangles in nanopores. So they're spread across the tree, but we do have a bit of a clustering over here. Now, we're going to be comparing ourselves with standard reference-based, standard variant callers like STAN tools. So we need reference genomes for those. So what we did is we used two methods to choose references. First, we used MASH. So for each of our samples, the gray triangles, we chose the MASH closest genome from RECOMB-Seq for each of them. So that's in red. And for each of them, we also chose, one genome per clade in purple. And for those who are interested, Shigella is here in the star. Shigella really is E. coli. Okay, right. So we find, how are we going to find all the true SNPs between the 20 genomes? Well, we do all pairwise alignments using NUCMA between the 20 genomes to generate truth SNPs. We only take SNPs inside, nicely aligned regions. And then you have to decide if a SNP that we find between genomes 1 and 2 is the same as a SNP between genomes 3 and 4. Okay, so this is annoying. We have to, in order to do validation, we need to find another method that does the same thing as Pandora, but which is absolutely definitely right in order to provide a validation, sort of golden truth. So how we do this? Well, we create a new graph. We basically deduplicate the pairwise SNPs. So we create a new graph. Each node is a SNP between two genomes. So this TA SNP is one node, and this TA SNP is another node. Okay, and we join two nodes if they share a genome or position. In other words, the node for this AT SNP and the node for this one are joined because they share this genome and this position and that allele. And then you find connected components within this graph, and these are equivalence classes of pairwise SNPs. When you do that, you find about 685, you find some edge cases which we throw out because we want high quality SNPs, and you end up with 685,000 differences between our 20 genomes. That's a lot, because we're basically comparing the whole thing. And how do we decide if a call is correct? So what I've done there is I found the true differences between the SNPs. Now I've got a VCF of calls from Pandora or SAN tools or whatever. How do I decide if it's right? Well, here's the VCF reference, and here's the SNP that the caller has found. We can make a probe out of the SNP, allele, and flanking sequence, and we can map it to the truth ref because we've got high quality assemblies for the samples. And then we can, having done that, you can look, and if the SNP is there, or the allele, the variant is there, and it matches, then that's great. I don't care about mismatches in the flanks, as long as the mapping of this thing's got quite a, I think we've got at least 100 bases on either side. Mismatches in the flank we're not so worried about. That should give you a true positive. But if there's a mismatch in the actual allele, then it's a false positive. And then I'm going to drag you through these details. How do I decide, how do I measure my error rate? So if the variant is ACAGT going to TCTGA, so that's 3 SNPs, T, T, and A. If Pandora called it as TCTGC, so we differ at the last base, we've got one of those 3 SNPs wrong. So there's 2 ways of measuring this error rate. Is my error rate 1 out of 3 because there's 1 SNP out of 3? Or is it 1 out of 5 because I've got 1 base out of 5 correct? Okay, you could do both. If you are only ever looking at non-ref positions, then there's an argument for just looking at the SNPs only. Because we're making calls at every position, we're making ref and alt calls, we want to evaluate everything. So we chose option 2. And anyway, we're applying the same formula to both us and competitor benchmarked tools, so it should be at least fair. Okay, so this is what the results look like for Pandora with 100x of Illumina on those 20 samples. So on the x-axis, I've got a stupid thing covering my screen. Along the x-axis, we've got the error rate. So in other words, what's the proportion of you can think of it as what's the proportion of the VCF records that are wrong using that error measurement that we described before. And so on the y-axis is what proportion of the 685,000 variants we can call. So we've got multiple lines here. Each line represents a different filtering that's been applied to Pandora. I've got 3 independent filters: a coverage filter, 15x depth minimum, a strand filter, I want 20% of reads on each strand, and a gap filter. I want at least 50% of the k-mers on an allele to have coverage bigger than expected depth over 10. And those 3 filters all independently improve the precision. So you can see these lines moving further and further to the left. And the reason there's a line is you can turn the dial on the genotype confidence for your SNPs. And the more confident you want the genotype to be, the further down the curve you move. You get more precise, but you have a penalty in recall. Okay, so that's sort of fine. That seems, I don't know. It seems fine. I don't know. Is 78.8% recall good or bad? Is 0.5% error rate good or bad? Let's have a look at, I'll come back to that. Let's have a look at how much local assembly is added to the whole thing. If I ran Pandora without the local assembly and then with orange and green, you can see there's a 3% increase in recall caused by the local assembly. And then if I compare with standard SNP calling on the same data, on the left, oops, there we go, sorry. On the left, I've got results from Freebase, or Freebase wrapped inside something called SNPy, which is commonly used in bacterial genomics, and run with multiple references, or the references we chose before. And you can see each reference sort of tops out at a different level. And then SAMtools is over here. Pandora is in the middle, so we get slightly higher recall than the other guys. But I mean, there's what feels like a painfully large gap in precision here. Like Freebase really manages to get very, very few errors. Okay, and yes, so I've already said that that's SAMtools. So this is an outstanding issue, and it will remain outstanding until the end of the talk, essentially. I don't like the fact that our Illumina error rate is worse than Freebase, and we're in the middle of debugging it. So 1/9 of our errors are due to Pandora false positively saying a gene is present. And that's actually fixable. There are multiple ways to fix that, including simple threshold filters. But the second thing is that false positives, we only discovered this week, the false positives are enriched at the ends of the genes, and some of our input multiple sequence alignments, which we built from, they have sort of some truncated and wrong alleles in various places. So we're going to have to try, we're going to have to curate that. That's my hope for what will fix that, but I have no idea if it will work yet. So that's just airing our dirty laundry, essentially. So, okay, as an audience member, 37 minutes into the call, I've dragged you through all this stuff, and then, the recall's not that much higher, and you've got a higher error rate than Freebase, and we've had Freebase since 2010. But why are we bothering with all this, right? What's it for? Well, if you remember, the whole point was to be able to access SNPs in the non-core genes. That's why we're doing this. So let's have a look at that. If I break it down by frequency, along the x-axis, I've got how many samples a SNP is in, or not just the SNP, but its locus. And on the y-axis, I've got recall of the truth SNPs. And the green lines are SAMtools, and the red lines are SNPy, which is Freebase. And there you can see the difference, because Pandora here, with and without de novo assembly component, is here. So this is why we wrote Pandora, okay? These are all variants inside accessory genes of E. coli. These are all things that we're super interested in biologically, and they're not really accessible. It doesn't matter which reference genome you use, which is what I've said here. If I replot this as actual counts, because this plot that I was showing you, sorry to switch. This plot that I was showing here was recall as a proportion. If I change it to actual counts of variants, then obviously there's not many, not obviously, but we know from our frequency distribution, there's not many genes in the middle, and there's more on the left, and there's more on the right. And so here we can see there's a gap of more than 10,000 SNPs, okay, that we can access in genes that only occur in pairs of samples. And this gap can only get bigger as the set of samples gets bigger. So this is almost, I could almost go home now. This is why we did it. And then if we do it on nanopore, I go back to the same plot where we look at accuracy. It's the same idea, the same filters independently improve precision, and we end up with, again, about 78.5% recall and about 0.5% error rate. And then, you know, I can't see you, I can't hear you, but if I was you, I'd be saying, what the hell is this? How can the headline recall and error rates be essentially the same for nanopore and Illumina? This is bullshit, right? This is how is this possible? Okay, so let's think about that. Let's first plot them on the same plot. Nanopore on the left in orange and Illumina on the right in blue. There is a difference in the error rate. Okay, it's not enormous. Look at the scale on the x-axis. Okay, they're 0.45% to 0.5% or whatever. It's not massive, but it is there. How is that possible? A few things. First of all, remember the graph is a strong prior. Okay, it allows us to zoom in on a close approximation, which is unaffected by nanopore errors. There's no nanopore sequence in the mosaic. So when I do my first level approximation, I find the closest mosaic. The only way nanopore can fuck me up is if it has systematic errors, which have been seen before in my high quality graph. So that's the first step. The second thing is mapping long nanopore reads is easier. You know, they cover multiple genes, and that allows me to do better filtering of bad minimizers. And the third step is nanopore actually only does this well if we use methylation-aware base calling. That's the reason why the de novo assembly step adds recall for very little cost. And I'm going to say tilde0 because in the example I'm about to show you, there's zero cost literally. In some other samples, I've seen a marginal cost. So it depends on the data. So here's a different data, unfortunately, because now it's only 4 of the 20 samples. And in red, I've got the best mosaic found by Pandora with no local assembly based on methylation unaware normal base calling, okay? And when I go from the mosaic to adding de novo assembly, that's what this arrow takes us from the red to the blue. There's this x-axis movement is the increase in the error rate, okay? So that's the penalty you pay when you've used normal base calling. If you use methylation aware base calling, then you go from the green to the yellow. Okay, there's a really appreciable difference, at least in E. coli. And then if we look at Medaka, so this is the dodgiest slide of the whole talk. We only got these results yesterday. I think the results are okay. I think the calls are okay, but I think that it's possible that, like, if you added more filtering to Medaka, it would give you more sensible results. At the moment, it's got a massive 20x higher error rate. It has, you know, a perfectly good recall overall, but these error rates are not acceptable. So don't quote me on this. We're going to have to work on seeing whether there are more sensible filters to add to Medaka. But if you do, but I think the calls are not crazy. I mean, if you break it down by frequency, this plot, you know, it's exactly the same plot we had with all the other reference-based tools on Illumina. You see the same stuff. I think you know, I think it has worked. Okay, that's a bit of a rush. We're 3/4 of the way through the talk. I'm going to remind you the heroes of this: Rachel, Michael, Leandro, and Leah. And let's just think about what this all means. So we start off with this representation. We use minimizer DAGs, and we take a union of small graphs that's driven by biology. I know that there's going to be lots of rearrangement. I want to be able to access all of these different genes, and it allows massive parallelization. I construct from MSA to preserve more of the haplotype information and avoid blow up. And the minimizer graph allows us to map with nanopore and Illumina sort of transparently. And then single sample inference, we do that in 2 stages. We quasi-map, we don't use full graph alignment. We use dynamic programming to get a mosaic, so it's already a pretty good approximation, and then local assembly for some improvement to that. And then finally, multiple sample improvement comparison, we use dynamic programming on the sample paths to choose a VCF reference. That's one path per gene or intergenic region. Now, a consequence of the way we're doing things is that indexing is not a slow one-off offline process, because we need to augment the graph with novel alleles from all our samples in the middle of our workflow. And that's the slowest bit of our workflow at the moment. Now, I've said this before, if you choose a standard genome as your VCF reference in advance, you immediately inherit hard bias. So we need a reference to make the VCF output possible. It's about representation, it's about description, it's about communication, but it's not part of our discovery process. So by using our approach, we remove the hard reference bias and you maximize the clarity of the variants, more SNPs and less long alleles. And just a reminder, you can't escape biology. You can't escape this frequency curve. No linear reference is enough, but no pan-genome reference graph is either enough because it's also built from a fixed panel. And when you add more samples, every new genome you see in E. coli, you find genes you've never seen before. So it's less biased than a linear reference. It's not perfect IMO, that's fine. Okay, we need it. And there's a question of whether that dynamic programming to choose a VCF reference actually helped. Here's a plot. This is how often a gene occurs in the population on the x-axis. Oh, and on the y-axis is the number of times in a sample that contains that gene is a perfect match to a VCF reference. It's edit distance 0. So how often is your reference perfectly the same as your sample? And so there's actually nothing to describe, okay? And you can see it happens sort of 1,500 times. Well, these are genes that only exist once in the population, and there's no SNPs there, but they're 1,500 times where we perfectly recapitulate the gene. And then over here, this number of times where in genes that occur twice, the reference is perfectly the same as a sample. And if you compare that with these 4 references, which are 4 references from the 4 major clades of our data set, it happens far more often in our using our reference. So we've basically managed to find a way to move closer to our samples. If I just ask, instead of being a perfect match, to be within 1%, so that most of the things you were describing should be SNPs, then again, it's far more often. But you'll notice that I seem to be cheating. I've restricted to gene counts less than 9. If I zoom out and say, does it apply to the really common genes? Well, no, it doesn't. So over here, once I get over here, Pandora's reference is over here in black, and you've got some of these other references are now doing better over here on the right. So what's that about? That is about population structure. So we should have built our E. coli pan-RG from a well-sampled genomes from across the phylogeny and geography and environment and host. We could have used the tree to select samples from which to build our graph. But instead, we just took 300 genomes, 350 actually, from RECOMB-Seq. And what happens is, if you choose references within here, because there are many samples in there, you're more likely to do well. So that's nearly the end. I should point out some limitations of where we are at the moment. Our multiple sequence to graph construction algorithm is in Python. That augment step is slow, that we're about to convert that to C++. We also have code which uses nanopore reads to collect gene order information, and that will come, but not in the immediate future. And in conclusion, well, variant calling, I think in bacterial genomes is quite different to typical eukaryotes. You can't escape this frequency distribution of shared chunks of genome. So it means there's a huge range of variation you can't normally access. So this kind of approach, I think, allows you to get much more excessive variation than a linear reference would. Still a work in progress. Still, I don't think I would say it works, but I wouldn't recommend people use it just yet. But the first fully functional release is not far away. The preprint won't be long. I've been saying that for a little while. That's the GitHub URL. And thank you. There's some people I should thank. In particular, these guys helped us getting the MSAs for our input data. These guys all helped with accessing E. coli samples, which we use for our evaluation data set. And Rob and Fatima welcomed Rachel at an early stage of the project and were really helpful with discussions about how we were going to develop this. And thank you. I'll hand over.